When you make little compromises in your ethics and your sense of what's right and wrong as you're proceeding, what happens is you're basically giving away a little part of your soul. And as you grow older, if you've done this, made these little compromises, you end up hollowed out. And over time, it becomes easier to justify amoral decisions. And it can lead you to a very dark place. And there are those, particularly in big laboratories, that have gone down that path. It's easy to fall victim to kind of the ends justify the means logic.
I've wanted to tell this story since the term mRNA entered the public's vocabulary during COVID. At that time, we started hearing a lot about mRNA technology. To the general public, that meant a new form of vaccines and a way out of the mess we all found ourselves in. But to molecular biologists such as myself, it meant adapting a technology known as transfections from small-scale benchtop experiments to population-wide human rollout with very little public education in between. What concerned me the most wasn't whether this technology would be safe and effective, because of course the data had not been borne out yet, but it was the silence from my biologist colleagues who are so intimately familiar with this powerful technology and its limitations. This episode is not meant to sway you one way or another on the merits of mRNA as a vaccine, but instead I want to tell the story of one of the most important technologies in molecular biology, what it is, how it was invented, and how it ultimately became one of the biggest hot-button issues in biotech. And who better to speak with than Dr. Robert Malone, the pioneering inventor of mRNA transfection technology, a tool that changed molecular biology forever. What is transfection? Simply put, it's the method of delivering synthetic nucleic acids like mRNA or DNA into cells to study or alter gene expression. Before Malone's breakthrough in the late 1980s, we couldn't easily introduce foreign genes into cells. His innovation using lipid nanoparticles to ferry mRNA across cell membranes revolutionized everything, from dissecting the central dogma of biology to engineering bacteria and yeast to produce life-saving drugs like insulin. Without it, our understanding of cellular mechanics would be decades behind. In this episode, we'll start with the basics. What is mRNA, and why is it at the center of almost everything molecular biologists do? We'll hear the untold story of how Malone invented transfection at the famed Salk Institute. Then, we'll bridge to its real-world leap. mRNA vaccines is essentially population-scale transfections in humans, swapping genes for antigens to fight disease. Educating the public on this is crucial because mRNA isn't just history, it's the future of medicine, from personalized therapies to rapid pandemic response. Join me in vivo with Dr. Robert Malone on the untold story of mRNA. Hey, Dr. Malone, it's an absolute honor to speak with you today, not just for your courage in challenging the status quo, but also for your pioneering work in molecular biology, which is really hard to quantify in terms of the impact it's had on everything from research to drug development. Now, one of my main goals here today is to impart on the listeners how central your technology has become in what molecular biologists do day to day. And I also want to frame this as a story of foresight rather than hindsight, which is really easy to do when we look back at the COVID madness that we all went through. While we were in the depths of it, I noticed a really troubling phenomenon of silence from our molecular biology colleagues who are so intimately familiar with the mRNA platform that you're credited with inventing, familiar with its immense potential and its inherent pitfalls. Now, the reason why we're so familiar with your technology before anyone ever heard of mRNA vaccines is because we use it in the lab every single day. It's become completely indispensable. And in my opinion, it's one of the three great game changers in biology. You've got the first one being the ability to manipulate DNA with restriction enzymes, Western blots for detecting protein expression, and then there's the revolutionary technology you developed for delivering nucleic acids into cells to study gene function. Together, these tools allow us to unravel nearly every node of the central biology dogma, or the dogma of central biology, and without them, who knows where the corpus of biological knowledge would be. So I think it'd be best if we start with first principles, fundamentals. What is mRNA? What exactly is it, and why do we focus on it so much as molecular biologists when we do our research?
Well, that's a kind lead in, and thank you for that. I would add, in terms of transformational technologies that give rise to modern molecular biology, you mentioned Western blots, Southern and Northern blots also. Yes. Southern is the detection of using kind of a similar approach of DNA, Northern being RNA, of course, sequencing, and then, of course, Corey Mullis. Talk about a transformational concept.
Let's not forget.
I remember when that came out, in my mentor at the time, Inder Verma, was actually jealous. Why didn't I think of that? I could have got the Nobel Prize. I think Inder spent his whole life frustrated over the fact that he kind of enabled David Baltimore's Nobel Prize,
right,
because he did a lot of the heavy lifting for David in terms of characterization of reverse transcriptase. But all he got was a full professorship at the Salk Institute before he was 40. Poor boy. Yeah, look what he did with it. In any case, so thanks for that lead in. Now, what is mRNA? And just to frame this up, we're speaking to a lay audience. Even though you're a card-carrying molecular biologist, and I once was, maybe I still have a emeritus license as a molecular biologist. I don't know. But we have to simplify this, not dumb it down, but simplify it. So the metaphor that there's a kind of, how do you express these things to somebody that hasn't taken cell biology or biochemistry or even organic chemistry? And the way that I deal with it is I reach for two different metaphors that everybody understands. One is a fried egg, and the other being a metaphor for the cell. And the other is the knowledge that we all have right now about digital information systems, what we call computers. And so my wife doesn't like these metaphors, but it's the best I've come up with. So if we think about how digital information gets processed, you have to have a place that you store the information. And then you have to have a way to transmit that information down to technologies that operationalize it. You could think of it as robots. So one way to think about this whole space is you have sort of a hard drive that stores information and repeatedly makes it available for reading and processing. You have a technology that's necessary to transfer that information out into the area that it needs to be used. And then you have a series of technologies. These are all biologics that have evolved over time, or depending on how you want to look at it, the creator generated. But in any case, they exist, however they came to be. You have a kind of a biologic technology in the cell that takes that information and turns it into operational output that we call proteins. And RNA plays a central role in all of this. Of course, deoxyribonucleic acid, DNA, is a double-stranded storage form of information that, unlike in the digital world where we have zeros and ones, it's a binary system. In the case of molecular biology and biology, all biology that we know, organic biology, you basically have a system of 4 different information bits akin to zeros and ones, which in the case of DNA abbreviated as A, T, G, and C. And you need to have a way to ensure that this information maintains integrity through time and across generations. If you want to take Richard Dawkins' point of view, that we are all basically vehicles for transmitting DNA through time, you want to make sure that that DNA maintains integrity. So what are you going to do? Well, what has evolved or been created is a amazingly a robust system involving two copies of the DNA that wrap around each other in the notorious double helix of Rosalind Franklin, Watson, and Crick. And so that double-stranded DNA is akin to the storage on your hard drive. It maintains it not only within the cell in a way that has mostly integrity. We can go into the nuances of that, having to do with various forms of DNA parasites and recombination and other things. But basically, you have this storage form. And then somehow that storage form needs to get copied, that information, and moved out into the area where it actually does its business, gets turned into something functional. So now the fried egg metaphor. You can think of, everybody knows what a fried egg looks like. And so the yellow part of the fried egg, you can think of that as akin to the nucleus of a cell, which is where the DNA resides. And just if you imagine a fried egg, there is a membrane, a barrier that defines the yellow part from the egg white. That, in the case of the cell, is the nuclear membrane. And the nuclear membrane has specific holes or pores, and it actively facilitates and controls what information can get in and out, what molecules can get in and out of that protected space where the DNA resides. And one of those molecules is RNA. And it's not just any RNA. There are multiple different forms of RNA that are copied off of a DNA template. And these include various structural RNAs, as well as a message ribonucleic acid. So mRNA is messenger ribonucleic acid. It's a single-stranded copy that gets complicated. It's processed. It's not necessarily collinear with the DNA, but it starts out collinear as a copy of the DNA, and then it gets processed in various ways and exported. And it goes out into the cytoplasm in the fried egg, that's the egg white. And there it encounters literally a little nano machine, a nano-robot, that takes that RNA like a ticker tape, and it runs that RNA through a kind of a pore or a groove in something called a ribosome, which is the little nano-robot that makes protein based on the RNA code. And so what about the building blocks of the protein? So that just to go back just a step, in that metaphor of the little nano-robot that takes the RNA, this is not meant to be sexual, takes the RNA that's feeding through that groove and is being used to build the protein, that the scaffold for that very structure is another kind of RNA, okay? Ribosomal RNA. So we have messenger RNA, we have ribosomal RNA, and then we've got, well, how are you going to get the subunits of the protein, these amino acids? How are you going to kind of connect those amino acids in a structured way into this growing molecule that's based on a RNA template? Use the same basic biochemistry of pair bonding that is involved in the creation of the double helix and the transcription or making of the RNA, which is basically a copy. Use that same basic biochemical technology to couple. So you have an amino acid that is tagged onto another kind of RNA, transfer RNA, that has a 3-base connector socket you can think of. And so as you have this ribosome, that's this thing, composed of ribosomal RNA, it's the little nano-robot, and you have this message of RNA that feeds through it, that message is read 3 bases at a time, 3 of these letters at a time, by a transfer RNA that comes in, is attached to an amino acid, reads those 3 bases, says, oh, this is where I'm supposed to be. And then there's a biochemical connection reaction. And that gives rise to proteins, which is kind of the functional molecule that builds everything else in the cell, from lipids through enzymatic reactions, lipids, carbohydrates, all the other kind of stuff that has to go on in a cell all gets built. And by the way, so does the DNA. It comes back. The DNA gets replicated using proteins also. So that's my metaphor. I hope that made sense to your audience. And it's this whole mRNA is thrown about as an acronym that confuses people. It's really simple. And the M stands for messenger. R stands for ribonucleic acid, which is just a string of biochemical linked subunits that form a linear polymer. But in RNA, it's a little weird. Here's a nuance. RNA folds up on itself. And so you can't, it's nice, you can look at the pictures in the textbook that an RNA is just, mRNA is just a string. That is a gross simplification. It turns out, and this is kind of the essence of the RNA world, is that this molecule is super complex. It is wicked hard to predict what its actual structure is, because this folding and assembly is very much a function of the environmental conditions under which it exists. And it's very dynamic, and it's influenced by things, this is a read ahead, like the presence of pseudouridine molecules, which are modified bases within the RNA that are produced by a protein enzyme acting on the RNA that affect how it folds, how it functions, how it interacts with the ribosome, and all this other stuff. And of course, pseudouridine is a key property that was introduced by Karikal and Weissman on top of the technology that I was involved in creating back in the late 80s.
Now I want to, I would add one thing, the semiotic nature of what you were describing there. Essentially, you're describing what we call translation. And a good thing, I always impart this on my students, is that in biology, science in general, but in biology, there's a certain language that we use, and there's a reason why we use those languages. And no pun intended, we're using the terms translation because you're translating from one language to the other, which is from the language of DNA into the language of amino acids and proteins. And no matter how many times I hear somebody describe this process, I'm just blown away at its beauty, its simplicity, and its complexity, and its importance in almost everything to do with molecular biology. So, and I'm also very happy that you mentioned Rosalind Franklin, because she doesn't get her dues all that often.
Yeah. There's like 5 or I think maybe even 6 or more Nobel laureates involved in getting us to this very simple story that we just covered in the last 5 minutes.
And in my opinion, you should be one of them too, but we'll get to that later. So the next question would be this very routine thing that's happening inside of all of our cells, you and I right now, every cell, everywhere, almost, has become the central focus of what molecular biologists do for the most part. Why is that? Why is it that molecular biologists are so focused on studying mRNA and manipulating it with the technology that you've had a hand in inventing? We're going to get to the technology in a second, but why is it so at front of mind for molecular biologists?
Well, I like to say that we're all constrained based on the technology that we have available. Yes. So often we like to think of science as this Socratic pursuit of open-ended truth, but the fact is that it's very much driven by whatever technologies we happen to have available at the time. And also there's a certain kind of herd or group focus that often happens. And one of the gifts, let's say, of my youth was being at the Salk Institute in the late 80s and at the time, there was at least a half a dozen Nobel laureates, and Jonas was still there also. And notable among those was Francis Crick. By the way, here's a fun fact. His Peugeot had the license plate ATGC.
Perfect.
Who would have known? So he's passed away now. So these folks were there continuing their research and their focus, and it created an opportunity for a young person to become very aware of the history of how this had all happened. It's very much driven by technology, and there's kind of almost a vector, a thrust vector towards trying to crack this fundamental code of life. And a lot of this earlier work was very focused on what is the nature of the molecule that transmits information and controls information. What's the mechanics of that information transfer and replication, and then the mechanics of how it gets turned into protein, cracking the genetic code. All of that was driven that way. And a lot of those fundamental problems have been now resolved. We understand that you can pick up any molecular biology textbook, and it gets bigger all the time, or genes or whatever the latest is, and you can understand if you have the time and the patience, a lot of that stuff. The RNA world has been a bit of a mystery historically. It was very much a mystery at the time when I was working. It was really way out on the fringes to be asking these questions about. Matter of fact, I had a San Diego Supercomputer Center grant for modeling RNA structure, and the lesson learned from that is you can't really model RNA structure. It's too complicated. There's too much that goes on, and so it's kind of the next horizon. Yes. In understanding the fundamentals of how the cell works. And by extension, if you understand how the cell works, then you understand the fundamentals of how life works. And what it is kind of peeling back the layers of understanding or how this wonderful symphony operates. Mm-hmm. And in the RNA world has turned out to be extraordinarily complex. This molecule that has all these different functions is so susceptible to a variety of different types of modifications, manipulations, conditions, effects. It doesn't really lend itself to classical biochemistry because classical biochemistry is super reductionist. And RNA exists in the context of the cytoplasm and the nucleus and is very, very dependent on these fundamental principles, like, for instance, free water.
Right.
We have this concept, and kind of as you move into biology, and it's still a lot of pharmacology is grounded in this idea that the cell is a bag of water that happens to have things floating in it. And so the logic basically is, well, you can look at gradients of concentration and things like that, and understand what's going on. That's an old school way of thinking about the cell. The cell is really more like a gel. Mm-hmm. All the water is structured. And so then you have these. So it's structured because of the hydrogen bonding that happens with water, and it functions as a gel, which means that our ideas about aqueous concentrations coming from the world of biochemistry don't really apply. And so then you have these complications of, well, how do things actually move through this sludge, this structured water? What are the function of pores? How do you actually manage the transition? The cell uses this amazing symphony of membranes to compartmentalize and transfer material from one compartment to another compartment in the cell. It's incredibly complex. And RNA is at the core of that, but it doesn't lend itself. You can't take a microscope
Right.
and look at RNA really in the context of a cell. We don't have that technology. We do have the technology of PCR. We have the technology of sequencing. We have the technology of recombinant enzymes, you mentioned, CRISPR-Cas9 for recombination, targeted recombination. And so often what molecular biology has a tendency towards is to everybody kind of, there'll be a little technical breakthrough in the ability to detect some phenomena. Everybody will rush through that door, that window. It's like an Overton window. They'll rush through that window and explore everything they can around that space that's enabled by that tech. And RNA, it's been kind of a little bit resistant to that. We had Tom Check and company and the Ribosomes story. That turns out to be more complicated than we thought it was going to be. We have these really complicated nuances involving interference in RNA and in translation, and these short RNAs, and the complications of frame shifting. It just the more you look at it molecularly, the more complicated it gets. It's not getting simpler. It's getting more and more and more complicated. So I guess it's just a rich landscape for people to, publish manuscripts and get tenure and win grants and all that. But as an intellectual exercise, it's really deep.
Now, just to back you up on the fascinating nature of RNA, there's also this hypothesis of the RNA first world model. Not only is RNA an intermediary store of information, there are some examples, like, for example, ribosomes and ribozymes, that they themselves have catalytic function. They can do work. And that's one of the reasons why some hypothesize that actually it was RNA that was the first nucleic acid to exist prior to DNA.
I think there's a lot of merit to that.
It's wild.
Biochemically, it's a lot more straightforward.
It's a great rabbit hole if anybody's interested to go down that one. And so, where we sit, and thank you for that insight into the Salk Institute, because there's so many interesting stories that come out of that, and they have definitely contributed a lot to the world of molecular biology. And it's interesting to hear what it was like back in the day. Where we sit today, I said on the onset, molecular biologists are so intimately familiar with mRNA and your technology, which we'll get to in just a second. The reason why that is because, I get asked oftentimes by students, what do molecular biologists do day to day? And they do lots of different things, but a big proportion of them will spend their careers studying 1, 2, 3, a couple of genes and asking every possible question about that gene. What does it do? What protein does it produce? What happens to the gene when it's to the protein when the gene's mutated? How does it affect the cell? Does the cell grow quicker? Does it die quicker? Does it look different? Does it behave differently? And so the reason why we're so intimately familiar is because we have to have a way in which to take that mRNA message, which is coding for some gene of interest, call it gene X, force it into cells and see what happens. And this is a process that we call transfection. And it is one of the most routinely done technologies in any molecular biology lab. So I want to get to that now. Can you please define for us transfections, and how did you find yourself developing this technology that is just indispensable? And personally, for me,
okay, so I'll give you the real story.
Give me the real story, and let's define transfection for our listeners.
And I came out of I got a biochemistry degree at UC Davis. There was not a molecular biology degree at the time, and I was working. I got my subsidy as a student came through working in a laboratory of the laboratory of Dr. Robert Cardiff and Murray Gardner at the Department of Pathology at UC Davis, working on breast cancer models. And I didn't realize at the time what the significance was. Bob Cardiff had just finished a sabbatical with Bishop and Varmus. So Bishop and Varmus got the Nobel Prize for the oncogene hypothesis. So he'd come out of that, and he was working with a model of breast cancer in mice involving a retrovirus. Now, the central dogma of biology is that DNA makes RNA, RNA makes protein. Retroviruses have a way to back that up, and they go from DNA form to RNA form, and that's what I think Temin and Baltimore got the Nobel Prize for. So retroviruses have this weird property of going back and forth. And one of the things they do is they insert themselves into the genome of mammalian cells. And when they do that, they disrupt the genes associated with wherever they've stuck themselves in. And they have their own molecular switches, promoters, enhancers. And so they not only affect their biology as DNA parasites, which is what they are, but they also affect the surrounding genetic information because promoters, enhancers, particularly enhancers, act at a distance. They don't just act proximally to whatever the genetic sequence is. So that was the world that I found myself in. And as I said, it was molecular biology as a PhD or a bachelor's degree didn't exist at UC Davis at the time. It really was just emerging. So I went through in my training basically the fundamentals of microbiology, and it was at UC Davis. You may recall the plasmids, the Puck series,
yes,
plasmid at the University of California, like Puck 19 and 18,
PUC,
et cetera, all those pairs, and also the single-stranded versions of those that were used in phage. That was all created at UC Davis in about the same time frame that I was there. So it was like one of these, like at the Salk Institute, one of these weird, happened to be at the right place at the right time where a lot of things were happening, including kind of the origins of dideoxynucleotide sequencing. And so because of the training I took, I was trained in the more classical microbiology involving, among other things, the use of a bacterial virus called a phage, particularly phage lambda, to insert genetic information and modify bacteria. Okay, so that is called transduction. The process by which a cancer cell becomes a regular cell becomes a cancer cell has a related nomenclature, transformation. Now, the irony is that, or the complexity here for the student is that transformation is used in two different ways. There is a transforming principle for which the Nobel Prize was given that demonstrated that you could call, I think it was pneumococcus, that you could transform a pneumococcal bacterium from a rough form that was non-pathogenic to a smooth form that was pathogenic, and this goes back to World War II, as I recall, by applying DNA, is what it turns out to be, by applying a transforming principle that caused it to have this change. And so that was part of what gave rise to the awareness that DNA was this important information molecule, but it also gave rise to this nomenclature of transformation. And so it's used as transformation in a way is kind of synonymous with the word you just introduced, transfection. Transduction is the use of a virus, whether it's a bacterial virus or a retrovirus or recombinant retrovirus, like in gene therapy using a viral vector. Those are really transduction, and really transformation is the language that's often used there. And then there was this suite of technologies that were used back in the day for introducing intentionally, in a much more facile way, in a much more efficient way, small fragments of DNA into cells, whether they're bacterial cells or mammalian cells. And that came to be called transfection, or infection, it's a derivative, right? It's you're infecting by transferring a material, but it's not really a virus that you're doing it with. So that was the nomenclature for transfection. And back when I was, so now we're getting to the story. Back in the 70s and 80s, this could be done through a couple of standard technologies. The leading one was by precipitating the DNA using calcium phosphate, so kind of the constituent of bone. And so you would make these really small precipitates, and there was an art in how small you could make the precipitate, how small you could make the crystals, because it seemed to work best if the crystals were really small. And then there was a whole art, it was kind of a little bit voodoo, of how you could treat cells, particularly bacterial cells, so that they would more readily take up this material, these small crystals. And that was called making them competent. And there's a whole art form around competent cells. You could buy competent cells from New England biolabs. It was a whole thing. You could look up in the Cold Spring Harbor manual, loosely bound manual of how to do stuff in molecular biology. I don't know if they still have it. They probably do. It's kind of like your field Bible about how to make competent cells for transformation. So that's calcium phosphate. And then there was another tech that involved the kind of clumping of DNA using a molecule called DEA dextrans, small little beads. And that was another way that you could get DNA into cells. And then there was the hot new technology at the time in the late 80s, which was electroporation, using pulsed electrical fields to basically cause pores to form in the surface of cells, and particularly in bacterial cells. So that became kind of the method of choice in many ways, much more efficient than using competent cells in HB101 to deliver molecular information into a bacterial cell. And then eventually, and I was kind of at the forefront of that, that's something that's overlooked, is the development of the use of that tech of pulsed electrical fields in migrating from use for bacterial cells to use in mammalian cells. And then I really was absolutely the pioneer in then using that in vivo for RNA and DNA. That's never mentioned in all this chatter about the RNA world. So then now let's get down to the business. So I was busy trying to do reverse genetics, is another term, using RNA as a way to ask questions about a fundamental problem confronting the world of gene therapy, and particularly retroviral vectors. At the time, retroviral vectors, based on, I mentioned Murray Gardner a while ago, a virus that Murray had identified back in the day in mice that had become the standard way for gene therapy, the idea that Ted Friedman of UC San Diego pioneered. This pediatrician came up with the idea, I think it's in the late 70s, that we could cure inborn errors of metabolism, ergo genetic diseases like muscular dystrophy, by transferring good genes into the bodies of children that had bad genes, that had inherited mutations. And so this very simplistic idea had been operationalized based on, remember I mentioned retroviruses that go from DNA into RNA and DNA back and forth, and then they insert themselves into the genome of mammalian cells. And so retroviral vectors had been developed in where you cut out a lot of the genetic information at the center of the retrovirus and put in your gene of interest, maintain those elements of the retrovirus that are necessary for it to be transferred into the genome, and then complement the missing genes using an engineered mammalian cell that we call a packaging line. So that was the kind of the origins of gene therapy and the leading tech at the time when I entered Inder's lab at UC San Diego and the Salk Institute. back in was 87. So Ender was one of the top labs, and Ted Friedman was the other one, really, worldwide in the use of retroviral vectors. That's why I went there. And so now my story makes a little more sense. I come from mouse memory tumor virus, retroviral vectors, fundamentals of molecular biology, gene transfer, vaccines, which was a major focus in that laboratory at UC Davis. And I decided once I got into an MD PhD program that what I really wanted to do was become a pediatrician and cure inborn errors of metabolism, follow the vision of Ted Friedman. And here's a fun little aside. I went to Ted and asked him to be in his laboratory, and he said, no, I won't take you in my laboratory to work on gene therapy because it doesn't work.
Interesting.
Retroviral vectors aren't working. But if you want to come in and be involved in creating an ordered cosmid library of one of the key human chromosomes, I'd be glad to take you on for that. And I said, no. I've had enough sequencing for my entire lifetime. I don't want to do that for the rest of my life. And of course, what I did was I missed out on the opportunity to be an early player in the Human Genome Project. So that's the way careers go. But I wanted to do gene therapy. So Ted wouldn't take me, so I went to Ender. I was advised not to go to Ender. It was a wickedly vicious, competitive postdoc lab of about 14 postdocs. And Ender was notoriously abusive. But, I was young, stupid, and ambitious. And so you go into a postdoc lab as the only graduate student, and historically, Ender would not take graduate students. You got to figure out a niche. Otherwise, you're just going to get crushed. Yeah. This is the reality, for your students. Be careful what you wish for. So I went into this insanely competitive laboratory for molecular biology and virology, which had, at the time, I'm going to just give a little more anecdotes. Renato Del Becco was still there. His laboratory was across the hall from Ender's lab. Renato, of course, got the Nobel Prize for the fundamentals of mammalian cell culture. And this is the laboratory that had trained David Baltimore, had trained Susumu Tanagawa, other Nobel Prize winners. So insanely competitive, cutting edge environment.
Right place, right time for you. Amazing.
There was a postdoc, the leading gene therapy postdoc at the time was a guy named Dinko Valerio. Dinko was way out on the edge. He had abandoned retroviral vectors, and he was pursuing a whole new vector-based gene transfer technology that was right on the cutting edge, adenoviruses. And so Dinko was the pioneer, really, in many ways, in the same laboratory with adenoviruses for gene therapy. And when he left the laboratory, he created a company. This is another aside. The company was called Crucell. Crucell was later sold to J&J, and it is the Crucell technology that gave rise to the J&J SARS-CoV-2 vaccine. So both of these vaccines that were marketed all came from Ender's lab originally. So here I am in this environment. I got to come up with something that is not going to just cause the surrounding postdocs to descend like vultures. And what I wanted to do was ask questions about how retroviruses got packaged, the RNA form of retroviruses, because we had some rudimentary information from reverse genetics and deletions about where the packaging sequence was for the various mouse-derived retroviruses. So this packaging sequence is at the 5' end, and it clearly we didn't fully understand how it worked. And so what I wanted to do was be able to create in vitro. And there was this hot new enzyme that had just come out, T7 RNA polymerase. Up until that time, everybody had been using SP6. And so there was kind of growing opportunities in RNA world But all of that was still being used just as a way to produce proteins using a lysate of red blood cells, erythrocyte lysates. So you could take a reticulocyte lysate. This is a nucleated red blood cell, and you could make a protein preparation of it, and then you could add synthetic RNA to it, and it would turn that into protein. It had the ribosomes and the tRNA and the other components you needed. So ATP. That was kind of the state of the art right then. And I reasoned, what could possibly go wrong? We can make RNA in vitro, and we have packaging cell lines. That's why I told you the story about packaging cell lines that complement the missing factors that are necessary to assemble a retrovirus. We have packaging cell lines. We can make RNA in vitro. We can basically engineer that RNA to have whatever sequence characteristics we want. And then all we got to do is get the synthetic RNA into the cells. What could possibly go wrong? It took me a stupid amount of time to come up with a transcription vector that would produce a retroviral RNA, and also to start to ask the questions about, well, how can I identify a system for delivery, what you would call transfection, into a mammalian cell that would be sufficiently efficient that I could take these RNAs that I would make, put them into a packaging cell line, and get out a retrovirus, an infectious retrovirus. And this may sound crazy, but polio RNA is infectious. You can take purified polio RNA from a polio virus and put it on cultured cells, and you would get polio out. So the idea of RNA infectivity into cells and its use to produce an infectious virus, of course, it's infectious. That means it would be amplified in the cell culture. All you needed is a few polio viruses to originate from that, and then you could go and it would be amplified, and you could harvest them and all that good stuff. So what could possibly go wrong? Well, it turns out that DEA, dextran, and the precipitation using calcium phosphate with RNA is just insanely ineffective. It's just a fool's errand to try to get it to work. But I was banging my head against the wall trying to get this to work, because the whole idea of reverse genetics was dependent on this concept. And this interesting guy who's still at the Salk, a true genius, and a Nobel Prize also ran, named Tony Hunter, who keeps voluminous notes, was kind of a central hub of information in molecular biology at the time. Reviewer for Cell, all the top journals, etc. Worked at the other end of the hall from Inder. Inder and Tony often had kind of a frenemy relationship, let's say. And as other characters, Walter Eckert, it was an interesting time. So Tony one day comes to me and he says, "Hey, Robert, I'm recreating this is at the distance of decades." But Tony remembers that he still has notes about this, and he's not allowed to talk about it by the Salk. This relates to the patent issues that developed later on. But Tony came to me and he said, "Hey, Robert, there's this new tech Gordon Ringgold at Syntex has that you need to know about." Now, I think what really happened was that Tony reviewed Gordon's seminal paper on that Phil Feldner is on that involved the use of positively charged fats to deliver DNA. It came out of Syntex. Phil was working for Syntex at the time. And so Tony thought that it was Gordon, because I think Gordon was the senior author. And so he said, "You ought to call up this guy, Gordon Ringgold." and find out about this new technology that they've got at Syntex and see if it works for your RNA. So I call up Gordon, and Gordon says, oh, I'm not the guy you need to talk to. You need to talk to this guy, Phil Fogner. Now, here's the story of that, the origin of the use of cationic lipids for transferring nucleic acids. Okay, this is the story that Phil told me decades ago. He never tells the story anymore. It's all Phil the genius. But this is another one of these strange kind of circumstantial events that often is at the root of breakthroughs. Now, I mentioned that UC Davis was the source of the PUC plasmids and was very involved in the early days of transfection and expression of foreign proteins using bacterial plasmids in mammalian cells. And so a undergraduate student that had worked in that laboratory, that had developed those vectors, was doing, it's a woman, and I don't remember her name. I'm so sorry about that. It's like the Rosalind Franklin story, only a little bit smaller. So she was doing a industry internship at Syntex, which is kind of down the road in San Francisco Bay from UC Davis. And she found herself in the laboratory of Phil Fogner. Phil was assigned to developing ways to modify the charge on the surface of mammalian cells, which are generally negative. And Phil had come up with the use, Phil was a liposome specialist. There was some real core competence in the liposome world in the Bay Area, consequent to the development of the liposome company, the development of stealth liposomes in particular, their use for drug delivery, et cetera, et cetera. So Phil was part of that liposome world that had really developed to an advanced state within the Bay Area, Berkeley, UC San Francisco, et cetera, and Syntex. So Phil's job was, how can you take lipid technology and make it and adapt it for the purpose of modifying cell membrane surface charge? And so he took fusogenic lipids, known lipids that would insert themselves into the cytoplasmic membrane, that's the outside of the egg white in the metaphor, and modified them so that they would have a positive charge. In particular, he modified this using, now I'm going to get into chemistry a little bit, so follow me with this, stay patient. Quaternary amines are nitrogen-based molecules that have basically 4 carbon bonds, and it forces them into kind of a permanent positive charge. So quaternary amine is a permanent positive, as opposed to a tertiary amine. We'll come back to that because it's important, because it's tertiary amines that are used in the current formulations or the RNA in vivo delivery. And this is the work of the group up in University of British Columbia. So Phil was working with quaternary amine-based lipids, and this intern from Davis that had been messing about with transfection in the laboratories that had developed the Puck series, said to Phil, according to his story to me, when I asked him, how did this ever come about? She said to him, hey, DNA's negative. These things are positive. What happens when we mix them? Okay. And it was that simple. And that it is so fundamentally powerful, the combination of fusogenic lipids, self-assembling particles, in which you have this large, relatively speaking, polyanion, no negative charges all along the backbone of DNA molecule, and positively charged fats that will basically coat this. So it's not a liposome. That's a misnomer. Nanolipid particle is the right phrase. And it coats it, collapses it, creates a self-assembling particle, and this is key. A lot of folks like Judy White at UCSF mentioned her name prominently because she played a key role in my work, and it's acknowledged in the first PNAS paper, postdoc in her lab. So Judy White was banging her head against the idea of, well, we could take RNA, messenger RNA, and encapsulate it in standard liposomes, so these bags of fat, and put it into the lumen of those bags of fat. That's a traditional liposome. And use that to deliver that RNA. So that was going on in parallel. I didn't even know about Judy back in the day. That came out later. So other people were trying to encapsulate RNA, but the problem is the entrapment efficiency is horrible. It's 1%, maybe 2%, because the concentration of RNA inside the bag is the same as the concentration of RNA in the solution outside the bag. And so what you do is things like sonication, where you basically are breaking these liposomes open so that you can get a gradient, so you can get the RNA to flow into the lumen. And then you got to purify them away from the other stuff, the solution. And making RNA is pretty expensive business, particularly back then. And so you ended up with this horrible entrapment efficiency, and then they wouldn't deliver for beans anyhow. So the delivery tech didn't work. And that was their frustration in Judy White's lab, and others at UCSF carried that on. So that was traditional liposomology. But what happened in the kind of the lightning struck at Syntex story is that this aha moment where this person came from a laboratory that was already doing DNA transfer and said, hey, this is negative, that's positive. What happens if we put them together? They self-assemble into a particle. And if you formulate that particle so that you have an excess of positive charge from those quaternary means. Remember what I said about cell membranes? Cell membranes are negative. So you end up with this basically ionically driven interaction of, it's kind of like magnets. Plus likes minus, minus likes plus, and they stick. And when they stick, if you have fusogenic lipids in the formulation, these are lipids that are known to disrupt membranes and have a tendency, if they're brought into close approximation, to flip from one membrane into another membrane. An example of that is diallyl phosphatidylethanolamin, which is why you have dope in many of these cationic lipid formulations. In addition to, I actually got the check mark for, I think, being the first to also add cholesterol to increase the fluidity of these membranes. That's one small paper in the, in the history. But if you use these fusogenic lipids together with these ones that have increased the fluidity of the lipid nanoplex, and it hits the cell, then you get these fusion events having to do with lipid transfer. And then that seems to facilitate the aggregate getting across the cytoplasmic membrane. Not really clear. The electron micrographs are hard to kind of freeze whatever that event is. And it appears that it is some strange hybrid of a fusion event and something akin to endocytosis. Because these particles can end up in endosomes.
I think it's important to mention that the membrane that we're trying to pass the genetic material through, it itself is also made of lipids.
Sorry, assume that to be fundamental knowledge. That white egg has membranes that separate the yellow nucleus from the white cytoplasm, and separate the cytoplasm from the general extracellular milieu of fluids and everything else that's going on. And into that outer membrane is embedded a variety of proteins, and it is comprised of two layers. If my fingers are the hydrocarbon tails, of these lipids that comprise these two. It's the membrane is a bilayer structure, and it has at its core. So it's what is the word? Amphipathic.
Amphipathic. Yep.
At the core, it has a hydrocarbon, so it's oil-like. And at the outside, where my palm is charged molecules that are hydrophilic. They like water. And so you have water-liking molecules that also have water-disliking or hydrocarbon components, and the hydrocarbon components associated with each other, and the charged components associated with water. And so they will form a lipid bilayer, which is what's used. It's a fundamental. We talked about the origins of biology and the role of RNA and the primordial molecule and all that. Well, without a doubt, one of the fundamental breakthroughs that may involve lightning or aliens from outer space or God only knows what is the creation of an enablement of these small bags composed of lipid bilayers that allow the separation of one volume from the surrounding environment. And then that enables special magic to happen within the segregated area. So now we're getting into Brett Weinstein space. Yeah. So that's the one of the theories about the origins of life. But so you mentioned lipid bilayers, and I just had assumed that everybody understood.
Oh no, that's
that.
okay. That's okay. But it's important because the biochemistry is related. So what did it feel like the first time this worked?
Okay, so what happens is, getting back to the story, Tony tells me to talk to Gordon. Gordon tells me to talk to Phil. Phil says, we have tried really hard to get RNA delivery to work, and we can't get it to work. In the interim, what had happened was that Inder sent me off to this laboratory at UCSF because, well, Robert, you can't get RNA delivery to work. You ought to go to a real laboratory that's doing this kind of work. And I know of one at UCSF, and it's the laboratory of Judy White. So I go up and I spend time in Judy White's lab, and I meet a postdoc, and Pablo, I forget his last name, I apologize, who had advanced the use of chloramphenicol acetyltransferase open reading frame. So CAT was the technology that everybody used for detecting gene expression back in the day. And it's a nasty business. It involves radioactivity and thin layer chromatography in order to create this biochemical reaction that you mentioned before, Western blots. So this is thin layer chromatography that would allow you to separate and show that a biochemical reaction had occurred, the acetylation of chloramphenicol via chloramphenicol acetyltransferase of a precursor radioactive molecule. So nasty business. You can't do many CAT assays. It's a big experiment to do a replicate of a 8-lane TLC plate. So that was the tech that was being used in Judy White's lab, was the production of a mRNA that encoded chloramphenicol acetyltransferase. And the thing that had made that go forward for them in that laboratory is that they had used a 5' and 3' untranslated region for the RNA derived from Xanthopus beta-globin, as I recall, which is a very stable, highly translated RNA. And so what was done is to take those elements and put them into a cassette vector that would produce by SP6 a chloramphenicol acetyltransferase transcript flanked by the Xenopus beta globin 5' and 3' and translated regions that had a little short poly A tail tag to it because that's all you could get because the larger poly A tail is unstable in a plasmid and it self-deletes. So I got that plasmid from them. And then I had been, my first rotation at UC San Diego had been with this guy named Suresh Subramani. And Suresh had just published the paper in Molecular Biology, as I recall, involving his pioneering work on a whole new reporter gene called luciferase. And I had been in the laboratory. Luciferase is what made all this work. That's the honest truth. I would not have a career if it wasn't for luciferase, because basically it allowed you to now go and do at pennies and in minutes what it used to take hundreds of dollars and days in radioactivity and film exposure and blah, 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 blah with thin layer chromatography to do a CAT assay. So I took, I think Pablo is his name. I took his vector with the beta globin, pulled that out, dropped in luciferase, put it in a T7 context, and that became the workhorse. And I'd done that and demonstrated that I could produce luciferase RNA. And that had been the tech that I had used, the vector that I'd used with the DEA dextran and calcium phosphate experiments, which just gave me just tiniest little signal over background. Luciferase is so insanely sensitive. It's like linear over 5 logs. And so you can detect the tiniest amount of luciferase, this is like femtogram levels in a luminometer because it involves the production of photons that are very sensitively detected.
Right. So this is an enzyme that will produce a reddish glow of photons that you can detect.
Yeah, red green. It's the enzyme that makes it so that firefly tails glow.
Go. Perfect. And you were using this to show that you could get RNA into the cells that could then make this protein.
But you had to have something that was sensitive enough that you could do the reporter gene assay. And so luciferase, so you had the combination. Luciferase is just so much more sensitive than chloramphenicol acetyltransferase and so much more reproducible and linear over 5 logs.
Safe and cheap.
With CAT, you would have to do the assay and you'd blow it out. You'd have to redilute it and blah, 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 blah. Okay, luciferase, you could just do the assay and you had this 5 log linear range and what's not to like. So that was the platform. And then coming off of Judy White's lab, and then Phil said, we can't get RNA transfection to work here at Syntex. Maybe you can get it to work because you've done these other things. Well, the reason was because they hadn't done the heavy lifting of coming up with a reproducible system that was sensitive like this to be able to detect whether or not it was working.
Interesting.
So Phil, at that point in time, I write an invention disclosure about the use of RNA as a drug, which was totally heretical. Remember, I'm in a retroviral vector laboratory where the cutting edge is adenovirus vector, and I'm way out on the limb talking about the potential for using mRNA for gene therapy. Now, a reporter that I know from the New York Times, who's really buddy with Inder, another Indian, Purva is her name, talked to Inder about these days the other day. And he acknowledged that nobody was talking about doing this kind of stuff until I started doing it. So thanks, Inder. But that and $10 will get you a Starbucks now. So that's what happened was I filed an invention disclosure. I talked to Phil about this. Phil said, we can't get it to work at Syntex. We'll send you some vials of this magic juice that he called lipofectin. That was this quaternary amine compound DOTAP together with and it's ether linked, so it's very stable. It won't degrade in cells, which is a bad thing. together with diallyl phosphatidylethanolamine, which was a fusogenic lipid, so the positively charged lipid with a fusogenic lipid, formulated as liposomes, sort of, in a clear vial. So he sent that stuff to me, like 8 of them or something like that, with an NDA, and it was like grease lightning.
Mm.
It just put the two things together. This T7 RNA transcript with the Xenopus beta globin. And at that point, I was also doing there's another enzyme that will add a longer poly-A tail, and I was using this new product that New England Biolabs was selling. As I recall, no, it was Behringer. Behringer was manufacturing CAP analog.
Mm.
So all that went into the tube, and then I did a little mini spin column to purify. So you end up with about $10,000 back in the day of enzymes and substrates in one reaction tube, which, I'm an unfunded graduate student. I don't know how I managed to get that through without Ender being aware of how much money I was consuming. But that's that. And then you'd have like a MIG of pure RNA, and you could do a bunch of experiments with it. So A plus B, put it on cells. Oh my God. The luminometer just goes.
And there you go. And the rest was history. And you mentioned earlier, your career would have not been what it was without luciferase. And there are many scientists sitting on this side who would say their careers wouldn't exist had it not been for the invention of transfections as you described them. So it's, for me, super fascinating to hear this,
so that's the actual story with all the warts and the serendipity and everything, all that.
I'm glad you got a chance to get that out, because that needs to be heard. It's fascinating. So, so what that, just to put a bow on it, what that allows us to do now, sitting here in 2025, but even up, 30 years ago, is we can now study gene function. You can take any gene using the technology that Dr. Malone just described, force it into cells, hijack the machinery of the cells to have them make the proteins, and away you go for, you've got a project that can last you 10 years simply because of this ability.
Yeah, you can do reverse genetics. Right. I never got to solving the problem of packaging because this one little thing became so big. I can't tell you how crazy things went.
I could only imagine.
And then I was in an embryology lab, TAing an embryology lab, and I did what the hell? I have all these Xenopus embryos sitting around. And then we were doing the chicking the cup process for growing chick embryos. And I just, and this set off a firestorm eventually at UC Davis because I was doing unauthorized animal research. Of course, you don't need authorization to work with frogs with Xenophus laevis, but that was university politics. But suddenly I was with DNA expressing beta-galactosidase. I was producing Xenopus embryos that had taken up foreign genes, and so the folks that were involved in the embryology world people were, Robert, you have to collaborate with me. This is like the future of the world depends on it, and my next paper depends on it, and I could get funding for it. But it just, the greed. So I ended up with a nervous breakdown.
Fair.
Which is why I ended up. I passed my qualifying exams. Focused on the issues of 5' 3' interaction, which was way out on the edge at the time in RNA world. And these various structural elements, the role of the 5' and 3' in translated regions, et cetera, in translation. And then all of this, there's a whole nother story that I've talked about in other places about the politics that went on. And I just got caught in the crossfire between University of California lawyers, Salk Institute lawyers, my mentor, quote, Andy Verma, other investigators at UC San Diego, and I couldn't take it anymore.
Yeah, academics is such a weird place. I mean, you've got this such a fascinating story to tell in terms of creativity, human ingenuity, and then you've got greed, ego that comes in and stifles things.
The Bayh-Dole Act has, for all of its good intentions, has really distorted American science. So that was early on. I was trained in the Bayh-Dole. That's why I was filing invention disclosures. At the time, the Salk Institute under de Hoffman, who had been involved in the Manhattan Project, he was the leader. Inder took his place after he left or died. I think he died of AIDS. De Hoffman was trying to create Salk Institute Biologic Associates, which was their for-profit subsidiary of their not-profit institute. And so he blew a gasket when he heard about all this because Inder was collaborating with industry without getting authorization. It's just a bunch of ugly stuff went down. And I was, Dorothy, you're not in Kansas anymore. And all the things that you thought, I thought I was going to the ivory tower, the summit of academia at the Salk. And what I found out is it's just as petty and egregious as any other place.
Thank you for that story. That's absolutely fascinating for me. I hope others find it interesting as well. But for people who, like I said, use this technology day in and day out, it's kind of like, reading Genesis. And it's I thank you for that.
Well, this is what blew me away with Peter Marks and Paul Offit denying that these particles would deliver DNA and that it wasn't possible that the Kevin McKernan observation about DNA contamination in the RNA products could lead to delivery of short DNA fragments into cells when injected in vivo. The cationic lipids are agnostic. They don't care if they just care if you're a polyanion. And RNA is a polyanion, DNA is a polyanion. They will bind and assemble around any polyanion you want. And so if you have DNA fragment contamination in your RNA preparations, which is something I had to go to great lengths to do as well as I could to prove that there wasn't DNA that was kind of a key to that initial PNAS paper. Then it will deliver. And as you know, as a practicing molecular and cell biologist, short DNA fragments are highly recombinogenic.
Yes. So
which is a fancy way of saying they cause mutations through insertional mutagenesis, kind of like the old story at the start about how retroviral vectors can be used to disrupt genes and detect oncogenes and stuff like that.
Absolutely. And so this brings me to the crux of what I wanted to talk to you about. I've been wanting to talk to you about for many years now. And that is when we were going through all of the COVID madness, I felt like I was in the twilight zone because I've got colleagues, I've got friends who are molecular biologists.
You and Byron Bridle both were in the what the hell?
Yeah, we, as I said on the onset, molecular biologists are intimately familiar with this technology that you just described. We use it day in and day out, and all of a sudden we've got talk of mass production, huge scaling up of this technology to deliver mRNA as a means of producing an antigen.
Your fellow Canadian, Dr. Peter Cullis, gets the gold star for advancing this. And I knew Peter from back in the liposome days. Let me just give you another story. So I knew Peter from back in the day. And as this was breaking, he would still talk to me. He won't talk to me anymore. And I called, I don't know why, tongue in cheek, but I called him up. I'm sure Peter is got a very nice yacht or whatever now. He's got all the money in the world, and God bless him. He had been working in liposome technology development delivery for decades. And I called him up early on. I said, Peter, what's the nature of these formulations, and what is it that is making them specific, and why are they working in vivo? Because I could never get them to work efficiently in vivo. What they mostly did was trigger neutrophil influx. They're insanely inflammatory. And he said, well, we have a new molecule that we've engineered that is the cationic lipid component. It's a branch chain, much more elaborate than what was used back in the day, which are, dual chains, hydrocarbon chains. And it has a tertiary amine rather than a quaternary amine. Remember, I told you this was going to
come back. Yeah.
So a tertiary amine is very much pH dependent about whether or not it's going to be charged or not. And so it turned out that tertiary amines, he believes, were essential to in vivo activity, because one of the problems when you use this stuff in vivo is that it interacts with extracellular proteins and other things in the extracellular milieu, and it can inactivate the ability, this key property of needing to have a net positive charge to drive it to association with cell membranes. So he said, well, we tried, a zillion different cationic lipids, and that's true. I mean, the whole field did. I did in my laboratory.
We experimented with some ourselves as well.
Yeah. So he came up with a new cationic lipid structure. It was a tertiary amine. And the other breakthrough was that he formulated it with diallyl phosphatidylethanol. I'm sorry. Yeah, no, not dope. I'm sorry. With I'm blanking suddenly. The molecule that's used in car radiators.
Glycerol.
No. It's not glycerol. Oh God, I'm sorry. I'm blanking on this.
It'll come to you.
It's a short side chain hydrocarbon linked to a God damn it, can't remember the name of the molecule. It's a hydrophobic. Well, it's a hydrophilic molecule that is used in stealth liposomes. And it gets highly hydrated, and it'll come to me probably 10 minutes after we finish our broadcast. It's highly hydrated, and what it does is it creates, remember I was talking about structured water?
The gel.
So it creates a structured water shell around the lipid nanoparticle. And what he did, that's fundamental to stealth liposomes, which when they're hydrated like this, they will pass through the liver. And then they do something called passive targeting, where they will accumulate in leaky capillary beds. Leaky capillary beds being a key characteristic of inflammation and cancer. So that's stealth liposome technology. And he used a version of stealth liposome technology that instead of a longer anchor lipid that locks those molecules in, he used a short chain hydrocarbon that made it so that when you formulated the particles, they would not coalesce, which is a problem with cationic. You know this in the laboratory. You kind of often have to make up the cocktail and then use it right away, because otherwise it will aggregate and flocculate. Yes. And, it'll turn into large aggregates and they don't work anymore. And so his breakthrough was in particular to use these short chain hydrocarbon linkers so that in the vial, the products don't aggregate. And upon injection, that tends to falls off because it's it doesn't have as much of the hydrophobic bonding characteristics. And so it tends to fall off, and you end up with a period where you have an active particle that will still be driven to associate with cell membranes. And his assertion was that these advances resulted polyethylene glycol. Finally,
of course,
in my brain. So these are polyethylene glycol linked to a short chain hydrocarbon that are used in the formulation process in such a way that they remain bound in the vial. and then fall off at some point in time after injection, which is why if you have a polyethylene glycol sensitivity, you can get an allergic reaction to these things. That's one of the adverse events. And like when I got my second dose of Moderna, which was the one that triggered my adverse events, I got one of the bad batches. I could feel shortly after the injection a tingling in my fingers. That's this release and distribution of this cofactor molecule. But it's what Peter in his group came up with to kind of make this whole engine run. And then Peter asserted to me at the time that this is what enabled them to solve the distribution problem, which is another one of your questions. Didn't they know that this would go all over the body? And he had actively promoted in manuscripts, published manuscripts, that this combination had resulted in a particle that would only would remain within at the site of injection and in draining lymph nodes. Presumably, macrophage would acquire the particles at the site of injection, pull them into draining lymph nodes. In the case of humans, that would be your axillary nodes. This is based on a small number of mouse experiments. That was not supported by the actual data that Pfizer and Moderna generated early on in their preclinical packages that showed this stuff would go all over the place, which is a whole nother story. They used the least sensitive method for detecting their reporter gene, luciferase, which was whole body imaging. It turns out that you can administer these preparations. I did it, for years. I was kind of one of the pioneers in this area. You can administer your gene delivery preparation encoding luciferase into your favorite rodent, and then which has ATP necessary for the reaction. And then you can inject the substrate for luciferase, which is called luciferin, extracted from fireflies largely. And you'll have a light, you'll produce photons. And you can take your mouse and put it in a lightproof box, with a highly sensitive photomultiplier tube camera called a VIM camera. That's used like for satellite technology, spy cameras and stuff. And you'll detect the photons that are coming out through the mouse and hitting the camera lens. And you can get an image of a mouse that is glowing in various places. And you can appreciate this is not a very sensitive technology compared to like dissecting the mouse and lysing the organs, but that's not what they did. In order to detect the distribution and fool the FDA, what they did was they used a VIM camera, a light box set up, and they looked at whole body imaging. And then as Byron Bridle, your colleague there at Guelph, documented, not only did they use the least sensitive technology for looking at the biodistribution of the gene expression in the products, but then they edited the images to cut out the inconvenient parts. And that's what they submitted to the FDA. So that's kind of where all this led. So I'm sorry, Peter. It's now, as you know, documented. You were wrong. The formulation that you came up with all of its masterpiece of engineering and insight, did not result in a product that remained localized to the site of injection and the draining lymph nodes. It goes all over the body. And perhaps the hydrocarbon side chains need to be shorter. I don't know. I think that there are fundamentals to the tech that are going to be difficult or impossible to overcome. And probably, if you're going to ask me the question that I get asked from time to time, can this ever be made safe? My response is something along the lines of, well, pigs could fly if they only had wings, and who knows what's in the future. One of my former postdocs that came from the Karolinska, named Peter Berglund, has continued working in this area. And Peter asserts that he and the group that he's working with now have a new delivery technology that is safe and that bypasses all these problems. And so I don't know what the future holds, but we're not going to get around these fundamental issues of pseudouridine, frame shifting, the potential interference, and other fundamentals that reside in the nucleic acid part of this.
And
we may get around the toxicities and problems associated with the lipid nanoplex.
I'd like for you to, if you don't mind, to comment on two things that occurred to me all those years ago as I was watching Nobody Say Anything in terms of my colleagues. And one of the obvious ones is when you're working with DNA or RNA, is the variability of the product that you're making, and then the exaggeration of that variability when you start to scale it up even larger, right? It's one thing to do this on a benchtop, working with cells, running your research, but when it's time to scale this up in bioreactors, hundreds of thousands of liters at a time, I'd like to hear your comment on the issues there.
So, and this gets where the rubber hits the road is what's referred to as process 1 versus process 2, among other things. So the initial clinical trials, I think it's of the BioNTech Pfizer product, employed a manufacturing process in which the templates were produced using PCR. And that has its own inherent flaws in terms of integrity and so purity and identity of the template. But it turns out that's expensive and not very scalable.
Right.
And so although they ran the initial clinical trials using a PCR-based template to produce the RNA, as Kevin, I think, again, I'm going to mention, he's not a doctor, Kevin McKernan, who has his own company because he was at the core of a lot of the technological innovations that enabled the Human Genome Project, and he made a lot of money. And now he just gets to play and do whatever he wants to do. And among the things that he wanted to do was find out whether or not there are contaminants in these, quote, RNA preparations. And so he and some others, but I think Kevin gets the gold star as far as I'm concerned for tip of the spear, started sequencing the residual DNA present in the vials. And then as an experienced genome person, he ran some standardized software to see if he could reassemble those fragments because they're random after he'd sequenced them. And lo and behold, what he found was that what had happened was that they had started using bacterial plasmid DNA and for the production of the RNA. So they had gone back to using essentially identical tech to what I had used back in the day. And the same enzymes, same basic structure.
Benchtop stuff.
Except, notable exception, that they used what was called back then a shuttle vector. And these are bacterial plasmids that not only have now we're going to talk insider molecular biology wonky stuff. They not only had an origin of replication for the bacterial vector in bacteria, but they had an origin of replication from mammalian cells so that it would reproduce these circular DNAs, would be able to be amplified and manufactured once they were inside a mammalian cell. So that's a shuttle vector because it can go from bacteria to mammal cell and replicate itself in both environments because it had the switch to replicate in bacteria and the switch to replicate in mammalian cells. What was that switch? It was the simian virus 40 origin of replication.
Much talked about.
In addition to the SV40 origin of replication, it also included a promoter enhancer cassette associated with that SV40 ORI. It is not the entire SV40. It does not encode large T antigen.
No.
That means that it doesn't have the traditional SV40 pathway for tumorigenesis. Sorry to all of those that have gone down that rabbit hole.
Yeah, you hear people saying the SV40 virus is inside the... No, not the case at all.
It's just these little fragments, but it's enough. As we mentioned before, having an enhancer, which is a switch that acts at a distance to turn on other genes. A foreign enhancer, very powerful, one of the most powerful foreign enhancers known to molecular biology, that of SV40 that drives the large T antigen, is not a small thing. So it turns out that why Pfizer used a mammalian shuttle vector employing SV40 sequences makes absolutely no sense at all. You don't need that to produce RNA. If you were going to engineer, if you sat down tomorrow and said, hey, I'm going to build something that's going to allow me to make boatloads of mRNA, what are you going to do? And I want it to be stable. I want it to be, I can grow pounds of this stuff. And I want it to be stable. Well, how do you make a stable plasmid, which is what we're talking about, circular DNA, to produce the RNA as the template? How do you make it stable? Well, you make it as small as possible because you don't want a bunch of extra garbage in there that can yield recombination events that will make that more efficient for replication, because bacteria will cut out any junk that they don't need to do the thing.
101, plasmid 101.
Yep. And so Pfizer didn't do that. They appear to have taken an off-the-shelf, 30-year-old technology. This is a shuttle vector that is exactly akin to the shuttle vectors that existed in the 80s, and use that to incorporate the SARS-CoV-2 engineered spike protein coding sequence. And there was no reason to have that extra garbage in there, but they did. So that raises the question whether that was foreknowledge or intent, or was it incompetence? And it's really hard to distinguish those things.
Yeah.
So that's one form of contaminant that you have in there. And because it's so large, it absolutely will recombine and you won't necessarily end up with a homogeneous product. And then there is the nuances of transcription using the T7 RNA polymerase. And that is, the fidelity of that is imperfect. So you now have a synthetic in vitro system. So it's in the test tube. It doesn't have all of the cofactors that even phage T7 would have to ensure fidelity in the T7 RNA polymerase, nor do you have all the cofactors that would be present in a mammalian cell to ensure fidelity of transcription and translation. And so one of the things that happens, you don't have the bacterial factors, you don't have the phage factors, you don't have the mammalian factors. And so what you end up with is a really sloppy process. Mm-hmm. And among the things that happens there is that when you're using this double-stranded template to produce a single-stranded molecule, you end up with, I think the technical term is D-dimers, you end up with RNA-DNA hybrids because you don't have the enzyme present that normally would resolve those dimers, which are potentially a contaminant in regular transcription, which is the fancy word for making RNA out of DNA. These hybrids between a single-stranded DNA at the replication fork. So the DNA has to unwind just to make available one of the strands to generate the RNA off of. So that RNA that's generated can hybridize and produce a dimer, an RNA, a heterologous dimer, an RNA-DNA dimer, which the DNase 1 is typically used to degrade all the DNA after you do all this stuff because you want to get rid of it. So you don't transfect it. And that doesn't work perfectly, and that's why you end up with contaminants. But it doesn't resolve, it does not have the capability to digest these DNA-RNA hybrids. And so that's probably also driving the presence of the high-level contamination of the DNA. It's intrinsic, to the fact that they're doing this in vitro. And the truth is that back in the day when I wrote that PNAS paper, I probably had this kind of contamination also.
No doubt.
And the truth is, as I think about it, this is one of the things, is I would run those gels to show that I'd gotten rid of the DNA, and often they'd had some really faint bands that I kind of just waved my hand around and said, well, it's almost all RNA. But it's probably, I didn't know any of this stuff back then. So probably that initial paper, well, the RNA is the predominant component of what I was transfecting with those studies that showed I could get a linear dose response in, R values, et cetera, for shooting the line. Probably there was some DNA contamination and probably some DNA RNA hybrid contamination.
So
also.
since we're on the topic of DNA contamination, can you speak a little bit to the potential of integration? This is one of those things that is often talked about, hand waved, not to worry, not going to happen. What is it? Has it been shown? Is it possible? What kind of danger might it pose?
So folks have actually taken now these preparations in vitro, or in situ, I should say, in cell culture. And we have more nomenclature. When we say in vitro, generally we're talking about in a test tube. We say in situ, generally we're talking about in cultured cells in the world of cell biology and molecular biology. And in vivo refers to in humans or mice or critters, living things. So there's our nomenclature. So in situ experiments involving delivery, administration of the actual vaccine preparations. There are data now showing that integration of the DNA contamination can occur. Now, back in the day, long ago, there was a PNAS paper from the laboratory of Rudy Jaenisch in which he used HepG2 cells. This is very early on in the mRNA COVID vaccine story. So Rudy Jaenisch asserted that you could get reverse transcriptase. Remember, we talked about that before. The enzyme that makes a retrovirus work that can take DNA and turn it into RNA, or RNA and turn it back into DNA in particular. That's reverse transcriptase. So the backwards RNA to DNA. So Rudy asserted that you could deliver these RNAs into HepG2 cells, which is a here's another word, transformed. So a cancer-like liver cell preparation that has been passaged for decades. Mm-hmm. So it's a long way away from being a liver cancer and has acquired a lot of genetic mutations and adaptations for growing in cell culture. So HepG2 cells are notorious for expressing line elements, which are kind of like historic retrovirus-like elements that encode something akin to reverse transcriptase. So Rudy did some experiments, and he asserted that you could put these RNAs onto HepG2 cells and get reverse transcription and integration. And a lot of people latched onto that. Problem with that is that Rudy has a long history, I'm sorry to say, of artifactual discoveries having to do with integration. So, and furthermore, using HepG2 cells is a long way away from using anything close to a living mammal. So that was the story until fairly recently, when after Kevin did his work and showed this contamination, the people went back and started to say, well, we don't have to look for reverse transcription. We can just look for whether or not these DNA fragment contaminants might be getting into the nucleus and into the chromosomes. Mm-hmm. And there are studies now showing that that's the case. I don't know if any of them passed peer review yet. But as you know, there's a lot of ways to do this and detect this. It's kind of almost a first or second year graduate level study to ask whether or not this happens.
Very straightforward. Fish.
And a lot of different ways to skin that cat using PCR or others. So there are some that are asserting that they have detected integration in cancers that have arisen associated with patients that have received these products. So then that goes down to a whole nother rabbit hole of whether or not those cancers are really associated with the vaccine. Are they just serendipitous, spurious artifacts? Are they associated with the virus itself? And generally, the way that this is approached is through monoclonal antibody staining that is specific for spike, and then you also use monoclonals targeted with some sort of a biochemical marker reaction or a fluorescent tag that are detecting the presence of the surface. The nucleocapsid. So you do a combination nucleocapsid plus spike, differentially labeled antibodies, and if in the tumor you're seeing all the cells expressing spike but not nucleocapsid, then the logical conclusion is that you've had some sort of an integration event, or you have a clonal expansion. This goes back to the retrovirus days that I talked about. So this is what one would do back in the day with retroviral vectors trying to detect oncogenes, is you'd look for a tumor that had typically by Southern blot, a clear signal of the majority of the cells having the same genetic rearrangement. And then you'd go and hunt for what the surrounding genes were, because you could probe for the retrovirus and then see if the tumor gave a single band for that retrovirus, then you know that's a clonal integration event. And then you could start asking, you could move out from there and say, okay, what is in the region of that integration event that might be a tumor suppressor gene or a positively acting oncogene, under the assumption that the LTRs, that the promoter enhancers of the retrovirus, were turning on these surrounding genes. So that's how that went. And so that's kind of the basic idea behind detecting monoclonality in a tumor that's expressing spike but not nucleocapsid. Well, that would say that it's probably the vaccine, not the virus, because if it was the virus, then it would be both.
That's right.
And so having identified those kinds of phenotypes, then you go hunting. Do you see any sign of DNA associated with this plasmid sequence that Kevin has popularized? And then infer that you've had because if you see that, you typically would have to have, and you can do the sensitivity if you're using PCR, you'd be able to say, well, at least 10% of the cells have to have this integration event or whatever, have to have picked up this DNA fragment. And so there are some that are asserting that they have those data now. Again, very small n's, very anecdotal, hard science to do.
Yes.
As and it's clinical research, because you got to get permission, autopsy or postmortem or a biopsy from a solid tumor, typically, in order to even ask these questions. So it's going to be a long way coming before we can really definitively answer the question of whether you get integration. But from first principles, as you know, there is nothing more wickedly recombogenic than a short DNA fragment.
Mm-hmm. Absolutely.
It is the perfect storm. And one of the things that people don't track with, this is another one of the misunderstandings that many lay people have, is the belief that these RNAs are going into all of your cells.
Right.
The whole idea back in the day, we're talking 87, 88. I didn't tell you another part of the story of the origin of the RNA vaccine concept, The postdoc working next to me at the bench was a guy named Dan St. Louis. And he was asking questions about the use of retroviruses to confer a reporter gene into, or a effector gene into a mouse model. And what he found, he did a thing where he would make little implants of transfected cells and then put those into the mice. And he would find that they would produce protein for about 2 weeks, the recombinant protein, and then it would stop. Whole laboratory was perplexed because this is a gene expression laboratory. They were working on Phos and June, and it was all about, the other part of the lab was all about gene expression. So obviously, there was some gene expression silencing event that was going on that was inactivating the expression off of these retroviral vectors. Everybody agreed that was the case. Having been through 2 years of medical school, at that point in time, I opened up my medical textbooks and scratched my head. And what came out of that was, one ear out the other. What came out of that was, oh, dummy, this is an immune response. 2-week timeline. Okay. And then suddenly the whole logic of exogenous gene therapy, of the Ted Friedman concept, went right down the toilet bowl.
Right.
Because your body doesn't know that it's a good gene. It only knows that it's a different gene, your immune system, and it will reject it. So if you put in the muscular dystrophy gene and you have a really efficient way to deliver the muscular dystrophy gene into all the cells of your diaphragm, that's what kills these kids. For instance, those kids' immune systems aren't going to say, oh, goody, we finally got the right gene. They're going to say, no. This is something different from what I've seen before. I'm going to reject it and I'm going to attack it. Cellular immune response. And suddenly the gene therapy paradigm went right down the toilet. And the only applications since then that have worked at all have been delivery into immunoprivileged compartments or with immunosuppression, pretty much.
Right. Makes sense.
So that was the aha when this all happened was, okay, Robert, you got this whole new idea of using RNA as a drug, as an alternative gene therapy, and it has the advantage that it only lasts for a few hours. The RNA degrades. So you can treat it more like a drug. And if the patient has an adverse event, you just stop administering the drug. What's not to like? And what are you going to use it for? Because now the whole idea about gene therapy is kind of on the skids. Oh, I know. You can use it to produce an immune response. You can use it as a vaccine. Well, the thing about a vaccine is that you don't need very much antigen. So the fact that it's relatively inefficient suddenly becomes okay.
Right.
It's the entry-level application for using mRNA as a drug in an incredible, the cationic lipid system, for all that it is in vivo, is incredibly inefficient. You can spend, you can spend a stupid amount of hours at a cryomicrotome, cutting sections, trying to find a blue cell.
Right.
Or a luciferase expressing
cell. We're trying to generate stable colonies with transfection.
So, but in the body, the immune system amplifies that signal. So what people don't understand is they think that, all of my cells now have spike protein. No. Well, if they do, it's because it's binding in separately because it's in the blood. But that's an error in assumption. That the truth is these particles are insanely inefficient. We just got a lot of cells. And you're administering a heck of a lot of number of particles. And so you get some delivery,
right?
And then the immune system amplifies the signal. So detecting an integration event is going to be wicked hard. Detecting what cells are actually producing the spike protein is going to be, generally speaking, wicked hard. This whole idea that we've got gobs and gobs and gobs of this stuff infecting all our cells is just a false, it's a easily refuted hypothesis. But we still have enough, and this was the stunning thing to me, was when I ran the numbers early on, I could not, there was some data early on about the levels of spike protein being produced. And when I ran the numbers in terms of, whatever it was, picograms or femtograms per microliter or something was what was being measured of spike protein. When I calculated based on blood volume for a human, I was like, oh my God, this is producing a way beyond the level of any expectation I ever had for efficiency of a system. So credit to the Arcturus et al. University of British Columbia crew for a far more efficient system than anything that I ever imagined might be possible. But it's still incredibly inefficient. But if you take a highly potent biologically active protein like spike, you can amplify the immune response to it, and you can also produce a variety of adverse events associated with it. And then you combine it with this pseudouridine, for which the Nobel Prize was given. And that's an important one to unpack too. So Carrico and Weissman, I knew Katie Carrico. She objects to my using this term. I could say I coached her or mentored her or whatever. I gave her ideas. She called me early on and asked for some advice about how to get involved in the RNA world. And I introduced her to some people, and I talked to her about some of the key issues, invited her to a conference. And then this is all a decade after I'd done that stuff at the Salk. And her and Drew Weissman observed that a fundamental problem associated with this was the inflammatory response associated with the lipid nanoplex. And they inferred, and correctly, that a foreign RNA itself evokes an inflammatory response. This gets back to your fundamental question, along the lines of, can this ever be made safe and effective? So at the time, the science of pseudouridine, it's still in its infancy. It gets back to how complicated the mRNA world is. But the science of pseudouridine was really just breaking through. Just a few papers at the time when and one of the observations was that if pseudouridine is in RNA, it seems to downregulate the inflammatory response associated with foreign RNA. And so they said, bingo. We're going to take out the U in the reaction, AUGC, and add pseudo-U, synthetic pseudouridine, and incorporate pseudouridine randomly all the way through the backbone. And then we're going to test that. And they found that they got higher signal, presumably using luciferase. And Katie tells the story about how overjoyed they were when they ran the first experiment and showed that they were getting more signal. And so that was the aha. And the patent filed was for use for cancer. It had nothing to do with vaccines, by the way. And the thesis was that the pseudouridine was impacting on the inflammatory response to the foreign RNA. And so this was going to be the enabling concept. And Katie basically went on a national tour promoting this.
I recall.
And so, yeah, she's got an interesting background that I won't go into. But so it turns out that pseudouridine does a lot more than just impacting on inflammatory responses. And it seems to be a longer term impact on downregulating immune responses, which is kind of a yin and the yang thing as it relates to this whole world. But among other things, pseudouridine really impacts on RNA folding and on frame shifting, which is this process we were talking about. Let's see if I can get it back again. The ribosome is the machinery. The RNA is threading through it and getting translated. And I talked about the transfer RNAs that are attached to amino acids that come in and dock the RNA as it's moving through. They can kind of misdock, and that's called frame shifting, and get polymerized into the protein. But then if they do that, then from then on, the RNA is misread. It's shifted its reading frame instead of the one that it was supposed to be based on the starting codon to something else. And so then after that frame shifting event happens, what it produces is gibberish, unpredictable, up until it hits some random stop codon and the particle disassembles. So you end up with, when you add pseudouridine, and you end up with these frame shifting changes in the protein product. And the cell actually does that intentionally. It modulates in real cell biology. Pseudouridine is specifically placed at certain places in an RNA molecule after transcription, making RNA from DNA. There's a post-transcriptional modification process that places pseudouridine at certain places in the RNA to change how it folds. That folding can affect how efficiently it's translated. It can affect how long it lasts, how it stays before it's degraded. It affects a bunch of different things, and we only understand part of it because it's very RNA and context specific. And it seems that during pseudouridine modification may change with different cellular conditions, stress conditions, etc. So pseudouridine modification is used to control RNA translation. It's actually used to control frame shifting. It's used to control RNA stability. And it's used to control folding, this superstructure that RNA forms. So we end up with a logic that was promoted, a patent that was held by UPenn, where Carrico and Weissman are, that has generated, I think, billions of dollars for UPenn, that UPenn sold off to a holding company and is used in, just as the University of British Columbia tech is used in all of these products for the lipid nanoplex. The pseudouridine is used for the BioNTech and Moderna products, BioNTech becoming Pfizer. There was another company that was capitalized by a gentleman that you may have heard of before named Elon Musk, that did not use the pseudouridine tech. It had to do with licensing agreements, and they didn't think they needed it. And I think that was called CureVac. And early on in the cycle of the race to develop an RNA vaccine for COVID, that firm took a more methodical approach to dose optimization. And they produced an RNA that did not incorporate pseudouridine. So very much like the approach that I had used, giving rise to a much shorter half-life RNA. We now know that these pseudouridine modified RNAs, and it's not even natural pseudouridine, it's a synthetic version that they use, confer very long-term stability to the RNA. Still don't know how long, but instead of hours, it's certainly weeks, if not months, maybe years. So this other company didn't do that, and they started at a lower dose in their initial clinical trials, in their phase 1 clinical trials. And Moderna and Pfizer used a much higher dose, Moderna being the highest of all in their initial clinical trials. And not surprisingly, the CureVac antibody titers were lower
Yeah, makes sense.
than the other two. And so the conclusion was that the CureVac product didn't work as well because it didn't have pseudouridine, obviously. Well, actually, if you go back and look at the data and you adjust for dose, the antibody response is pretty similar. So the question comes up, do you really even need the pseudouridine, which is absolutely associated with these frame-shifting artifacts, the production of surreptitious proteins of unknown function that are probably inflammatory, the long half-life, et cetera, et cetera. And of course, this is the basis for the Nobel Prize. You were going to say something about that. And I'll give you a little story about that. So way back in the day, like in 91, I was invited to the laboratory of Dr. Peter Lillystrom at the Karolinska to talk about the work that I had done, because Peter was working together with his graduate student at the time, Peter Berglund, who then became my postdoc that I was mentioning earlier in the story. So Lillystrom invited me out to the Karolinska to talk about RNA delivery because he was working on, drumroll, rimshot, self-replicating RNA based on alpha viruses, which is, of course, the new tech that everybody is going to, as you can greatly reduce the dose. And what could possibly go wrong with a self-replicating RNA? That's tongue-in-cheek. So Peter invited me out, and I talked about what I had done. And we had maintained intermittent communication over the years. And about the time when I went on the Brett Weinstein podcast, there was this ramp up of PR, clearly designed to get the Nobel Prize for UPenn and Kariko and Weissman. And I, having been around the Nobel laureate world at the Salk, et cetera, I was very familiar with how these PR campaigns are run by people that want to get the prize. So I saw this playing out, and I called up Peter and I said, they're taking credit for my invention, and they're saying that Katie and Drew did this all by themselves. They were the original inventors. This, interviews on CNN and the New York Times, et cetera. I found this a little offensive. It's kind of stolen valor. And I said, Peter, they're going to, should I try to spin up some sort of a press campaign here? And he happened to be on the Nobel Prize committee at that time. And he said, Robert, don't worry about it. They've already reviewed, we're aware of this. They've been nominated. We've already reviewed that nomination, and their contributions don't rise to the level of meriting the award. And I've made everybody aware of what your role was in all this. And in fact, that year the award went to somebody completely different. And then the next year, a new committee came in that I've been told is very woke. There was major investments in the region around the Karolinska in RNA vaccine commercial development and donations to the Karolinska. And that year, Kariko and Weissman were awarded not for the technology, but for their role in enabling these vaccines. And the committee said that the reason why was because the reason they justified the award was they wanted to encourage more people worldwide to accept the product, and getting the basically seal of approval of the Nobel Prize Committee would encourage more people to accept the vaccine product. So they awarded it for the vaccine product and the role of Kariko and Weissman, which actually they didn't play much of any role in the development of the actual vaccines. As we now know, for instance, the Moderna product was engineered at the NIAID at the Vaccine Research Center. And a lot of this was actually done through subcontractors. But that's what happened. They're not going to give the Nobel Prize twice to the same topic. And you move on. And in many ways, it's if I had been the third wheel on that prize, then I would be dealing with the Robert. I would have even more people accusing me of being a mass murderer than already do.
Right.
And, yeah. So
well said.
It is what it is, and I make my living now from Substack and trying to influence policy, and I'm grateful for the occasional opportunity to talk about the good old days like you're doing here.
It's absolutely my pleasure.
Technology, but most people want to talk about the World Economic Forum and the Great Reset and the fact that there's no virus at all.
Right.
And all these other interesting theories.
Absolutely interesting, but sometimes you do have to bring it down to base principles. And one or two last things before I let you go. Since we're on the topic of award, you were awarded the Courage and Humanity Award from the Swiss Association Leben und Leben lassen, which is Live and Let Live, and you accepted that at the European Parliament, I believe.
Yeah, that was unexpected.
Unexpected, but how did that feel?
It was nice. That was an interesting moment in time. More important was the opportunity to speak once again in the European Parliament, and advocate for freedom and the rights of individuals and freedom from coercion, compulsion, and enticement, the rights to informed consent, and to speak about propaganda, psychological warfare, these other topics. It's fun, and I know you live in the technology world, and I once did, but now I find myself in a strange position where people want to listen to what I have to say about bigger issues involving public policy and health. And I think I can have much more of an impact there. The person that was just calling me is asking me to assist in some key wording for a State Department document. So it was lovely and wonderful to be a keynote speaker there and to speak about the importance of these fundamental principles and the importance of pushing back against the globalist agenda. And that was really what the award was for. It was not for any of this stuff that I did when I was 28 and full of ego. Mm-hmm. It was for the courage to confront what we're all encountering now. And unfortunately, you folks in Canada are experiencing the brunt of this kind of globalist agenda, I'm afraid.
Unfortunately so. And your that work that you do outside of the science is very inspiring. I'd love the opportunity to one day talk to you about that as well. But on that note, my last question for you, and this is a similar question that I ask all of my guests, but this is a little more poignant here. What would your advice to students be? Those students, scientists, researchers who find themselves studying or researching something perhaps controversial, and they don't know whether they should or should not pursue it. There's pressure from egos, from finances, from academia as a whole. What would your advice to that student be?
Oh boy. So of course you want to say, you have to stick and maintain your integrity and stick to your principles and fight for truth and justice. Put it this way. Both of my boys are computer science engineers. Nobody wanted anything to do with academia or medicine or science. Having vicariously experienced the lives of their parents. My wife has been key in everything that I do, was my laboratory manager for years, etc. So one of my sins, you could say, it was a sin of ambition and ego that I was warned about, was going into Ender's lab. The person that was assigned to guide me in my PhD program, Debbie Spector, who's a CMV specialist, came out of David Baltimore by way of Varmus Lab, as I recall. Hated Ender with a passion from their time together in David Baltimore's lab. Strongly warned me to not go to work for Ender. But I really wanted to do gene therapy. And I'd come to UC San Diego from Northwestern to do specifically that. And Ted had said no. So I paid a price for being so ambitious. I think as a graduate student, there's a lot to be said for working in a laboratory that is cutting edge in terms of its scientific technical skills, but maybe focused on an area that is less competitive, so that you build the skill set and have the time and bandwidth to learn your trade, because that's what it is. It's truth is, I learned how to do science from Bob Cardiff as an undergraduate. I was a total lab rat, and every single week it was stand and deliver. What are the controls? What's the positive control? What's the negative control? How do you interpret this? Blah, 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 blah. I did hundreds of Southern blots and, 20 or 30 Northerns, and I just was a mill in grinding that stuff out. And that's where I learned how to apply the scientific principle, the method of multiple working hypotheses, that kind of stuff. So don't bite off more than you can chew. If you are, I think, to be realistic, you have to be strategic. And you're asking somebody who is, as a graduate student or even as a postdoc, you're still learning your craft. You have almost no status. You're easily taken advantage of. And you have to be a little bit strategic, and you have to recognize that if you're going to go into a modern big laboratory environment, you are going to be subjected to competitive pressures that you may not be ready for. So better to choose some place that is going to be a little bit more nurturing. Look for a mentor that will mentor you, not one that will exploit you. And who takes pleasure in helping young people to grow up and succeed in science, and yet also has the credibility that that when they write you a letter of recommendation, it will open the doors for you that you need to have open, that together with your publications. I don't think you need to, when you're running into a breakthrough idea, it's easy to get caught up in the excitement about it. You don't need to disclose everything. You don't need to disclose all the implications. And you might want to just focus on getting the work done and getting the initial proof of concepts driven home, perhaps a little less spectacular way. I think one of the lessons over time for me that I talk about on podcasts all the time, when you make little compromises in your ethics, in your sense of what's right and wrong as you're proceeding, what happens is you're basically giving away a little part of your soul. And as you grow older, if you've done this, made these little compromises, you end up hollowed out. Over time, it becomes easier to justify amoral decisions. And it can lead you to a very dark place. And there are those, particularly in big laboratories, that have gone down that path. It's easy to fall victim to kind of the ends justify the means logic. And if you want a notable example of that, look into the history of Luc Montagnier and Bob Gallo and the discovery of HIV or LAV. The history of science is so we mentioned David Baltimore. Case can be made that David Baltimore really took credit for other people's work. That a lot of the Nobel laureate stories have a dark side. We mentioned Rosalind Franklin early on.
Very good example.
So be careful out there, and be a little bit guarded. And my big sin as a graduate student was ego. I really thought I was fast-tracked, MD, PhD. My GREs for biology were off charts. I just pegged the needle because I just come out of hardcore biochemistry at the cutting edge of the origins of molecular biology and 2 years of medical school. So I just nailed those GREs. And I thought I was the best thing since sliced bread and I could do anything. Maybe that's part of what allowed me to do what I did is I wasn't constrained by fear of what I could achieve. My mind was very open. But I also suffered from the errors of the young man who's highly ambitious, and so it's hard, keeping that ego in check, and taking time. There's another thing that I've had to learn over time. It's often necessary to go slow to go fast. And that's a paradox. It's worth contemplating. And it applies to the graduate student and the postdoc also, is going a little slower in your research, in checking the boxes, allowing yourself to question whether or not you have run down all the rabbit holes. You have examined the alternate hypotheses. And the other advice I would give that my mentor back in the day, Bob Cardiff, gave to me that has just served me so well over time is there's this paper that was first published in the late 1800s in Science magazine and then republished in the 1960s called The Method of Multiple Working Hypotheses. Get that paper, read that paper, understand that paper. because the key, the easy sin that is so predominant is that you become invested in, quote, your hypothesis,
your story,
and you will fit the data. It's a form of confirmation bias. You will fit the data to support your hypotheses that you have a sense of ownership.
Mm-hmm.
If you use the method of multiple working hypotheses, you become agnostic. You acknowledge that there's a broad range of hypotheses that may explain the phenomena, and your only job is to design experiments that can differentiate between those hypotheses. And then suddenly you become divorced from ownership of any one of them. You're just kind of an objective observer, looking at the landscape of possibilities and discriminating between those to finally get to the only hypothesis that fits the data. And that idea is an amazing engine for innovation. I've gone back to it again and again. The other thing that is super, I think I do have a publication history that shows, I don't have as many papers as some, it's only like 100, but a lot of them are pretty highly cited. And what enables that kind of breakthrough innovation is the method of multiple working hypotheses. And the other one is recognizing that outsiders can see things that you can't. You become so invested in the language and the logic of your particular domain that it becomes extremely difficult to imagine explanations for physical phenomena, which is what we're investigating in a very abstract, esoteric way, molecular biology. You can't see it. You can't go and mess about with it. It's like you can't drive a hammer, It's almost like theoretical physics. But outsiders are your friends. Don't reject them.
Mm-hmm.
Even though they don't know the right words. When you're talking to your wife or your friend or this other graduate student who's in another field or whatever the thing is, and you're talking about what you're doing and what your ideas are, and they say, yeah, but what about this? Didn't you think about that? This sounds like something else. And they may not use the right language. When you encounter outsiders as you become a principal investigator, you can use this actively. If you draw in people that are outside of your discipline, they can be an engine for innovation because they can see things that you can't see. And you need to listen to them really carefully because they won't have the right language. They're not part of the guild. So you need to be able to translate, like interacting with a chemist or a physicist. If you're a molecular biologist, you're like, oh, come on, you stand on this. They'll use the wrong words, but they often, even if they don't quite have the idea right, listening to them can lead you to seeing things differently that you weren't seeing before. So there's my gifts to the student and the postdoc. It all comes down to having an open mind, but being careful because it is an insanely competitive world out there.
I love it, Dr. Malone. What an absolute honor, again, on a personal level, for many reasons. I appreciate your time. You've been very generous. What's the best place that people can look you up and follow your work? Is it on X? And your website?
So the best place is actually Substack.
Substack, even better.
And you don't have to pay, if you're a graduate student or postdoc, you don't have the money anyhow. You don't have to pay for the monthly subscription. But what we do is those that do pay for the monthly subscription, so it's free, you can sign up, it comes to your email, or you can find it on X or Getter or Truth Social or Gab, whatever your flavor of the month is, at RW Malone MD. But the Substack is called Malone.news. It's easy to find. We've got about 350,000 subscribers, of which only a small fraction are paying. But if you do decide to pay for the monthly subscription, thank you. And it's what keeps my horses fed and everything else. And because I no longer have a career otherwise, If you do pay, then you can comment, and we have a very active chat comment community. And what that does is it keeps the trolls out.
Of course.
So they don't want to pay $5 a month. So Substack, and then as I mentioned, X, Gab, Getter, Truth Social, at RW Malone MD, and then there's the shameless self-promotion.
Beautiful.
The books.
Excellent book. I highly recommend it.
Cywar and what's the other one? I'm even blanking now on the other book I hardly read. Lies My Government Told Me, The Better Future Coming.
Wonderful. And we'll link all of that below. Thanks again, Dr. Malone. Much appreciated. Happy New Year to you and your wife and your horses.
Yeah. And good luck in Canada.
We'll need it.
Yeah.
Thank you, sir. In today's conversation with Dr. Malone, we traced mRNA from its fundamental role as biology's messenger, bridging DNA's blueprint to the tangible proteins it codes for, to Malone's groundbreaking 1980s experiments at the Salk Institute, where he was the first to use lipid nanoparticles to deliver nucleic acids into cells. This breakthrough revolutionized everything. Ectopic gene expression for basic research, microbial factories churning out therapeutics like insulin, and a deeper grasp of the central dogma. But looking back on this masterclass with Dr. Robert Malone, I'm struck by a profound irony. The very technology he pioneered, which is a cornerstone of modern molecular biology, became a global experiment in human-scale transfections. Yet, its well-known pitfalls were met with widespread silence from those who knew them best. It's hard to describe without sounding hyperbolic how central transfections are to molecular biology. I myself have performed thousands of them, and there are tens of thousands of scientists doing the same every single day. This is the divide between clinicians and scientists. Doctors are great at treating an underlying pathology, but it's the scientists who develop and use the tools on the daily that have a responsibility to educate not just doctors, but the general public who are eventually downstream of these innovations. In that regard, and with the foresight molecular biologists should have had, we failed. Now, that being said, this particular technology does have a profound potential to change medicine the way it changed molecular biology. But as Dr. Malone said, sometimes we must go slow to go fast. Malone's journey from inventor to whistleblower underscores the need for ethical foresight in science, reminding us that true progress demands vigilance, not just innovation. So here are the questions I'm carrying with me. If mRNA's flaws were so familiar in labs, what pressures silence expert voices during a crisis? How might addressing these pitfalls reshape future therapies? And when science bridges bench to bedside so swiftly, how do we safeguard trust without stifling discovery? Thank you, Dr. Malone, for your pioneering spirit and courageous efforts, and thank you for watching. Please like and subscribe, and I'll see you next time in Vivo.